Good morning. This is truly a wonderful meeting. I'm very happy to be here. And I apologize that there won't be any physics in this talk. So we will take a completely different approach, like actually look at through the lens of evolution of condensates across the tree of life. So the idea that coacervates, for example, negatively charged RNA and positively charged peptides forming coacervates could have contributed to the origin of life is a 100-year-old idea, actually. But if we look at modern cells, biomolecular condensates are extremely complex. So there are many different proteins inside. There is RNA and DNA inside. They can have different localizations and different compositions with different specificities. So how did this complexity evolve, and how does it bridge actually to simple coacervates and many things we also study in the test tube to what we observe in modern cells is an open question, and this is something that my lab is particularly interested in. Are condensates essential, or more specifically, which condensates are essential, which could also tell us about their biological function, and also how are they distributed across the tree of life, and what is their molecular composition, and how is this complexity, did this complexity evolve, and the specificity of proteins knowing which condensates they belong to, and can we just predict this all from their amino acid sequence? So this is the challenge. So where do we start? So we know that IDRs are common in condensates, so we can start studying IDRs. So intrinsically disordered regions or proteins were conceptualized around 25 years ago, and before they were largely ignored, mainly due to structural biology techniques, basically like invisible to X-ray crystallography. But it turns out that 33% of the human proteins contain actually these IDRs, and they play an important role. And this flexibility and transient structures can be important for multivalent binding and transient binding that turns out to be important for biomolecular condensates. So I think the IDP field was really excited about the discovery of biomolecular condensates because finally there was a function that you could assign to many of these IDRs that before didn't have that. And then the last breakthrough I want to mention is the development of AlphaFold, which basically solved the problem of predicting from a sequence of a protein its 3D structure. But it's only true for the ordered region. So right, this kind of spaghetti-like structure around the ordered domain is meaningless, what it exactly looks like. And in fact, the lack of a predicted high-confidence structure is a good predictor of an IDR. But it doesn't really bring us forward to look at these images. And here we would have to think about ensembles, and there's a whole community who is simulating these IDRs, and they're looking at their characteristics as ensembles. What we are doing is not this. So we're actually trying to mine evolutionary data and use the huge amount of sequencing data we have accessible now because of all these genome sequencing efforts, and what we can learn from these. So, for example, we can ask the question, what is similar? So we can use this homology-based annotation transfer that we have been doing for decades, actually, for ordered proteins, and annotate function this way, and find out which sequences are similar. And once we have a set of sequences, we know they are similar, we can also ask what is conserved between them, so what features we see them conserved, and assuming that what is conserved across millions of years of evolution is actually functional, this can give us hints of functional sites, for example, within IDRs. And if we just think about a universe of all proteins and the size of UniProt that collects these proteins, it's billions of sequences, right? So the sequence space, we heard it yesterday, just for a 20-mer is 500,000 sequences. So we can cluster these now for the ordered regions. So I'm like taking this beautiful image or visualization from Joanna Pereira's paper when she looked at structural similarity between the ordered domains that we can predict. And we can see clusters that are similar structures. We can assume they have the same function. We can do this type of visualization for sequence similarity and find protein families and annotate, for example, function this way. But for the disordered sequences, we don't have such a visualization or such a way to actually compare them. And very, very few proteins are actually annotated with a function. This number comes from DISPRO, that's this disordered protein database. It's basically very low. And the reason for this is because not only AlphaFold is ineffective for these sequences, but also alignments. And alignments are basically the first step that you have to do if you want to do evolutionary comparisons, taking sequences from different species, comparing them. This works very well in this, just an example of this alpha crystalline domain of this heat shock protein. But for the two terminal regions, the IDRs, there are gaps. There's almost nothing conserved, often only short motifs conserved or the amino acid composition. So to break free of alignments and try to do this also for IDRs to find similar regions, what is so-called unalignable sequences, so anything that's not alignable, so it doesn't have to be disordered. We developed a tool that's called SHARC, that stands for Similarity Homology Assessment by Relating K-mers. And it comes in two flavors, so you can do SHARC dive to find homologous sequences, and SHARC capture to find actually the conserved regions within the sequence. And Willis, my PhD student who developed this tool, loves sharks. So he was very happy about the name. So just very briefly to explain what the algorithm does. So if you compare two sequences, which would have two motifs, in this case it are swapped, you wouldn't be able to align them and find both of them similar, right? But you can break down the sequence into k-mers, showing an example of K4, where you look at all potential k-mers between these two sequences and compare the composition of these k-mers, but also taking into account the physical-chemical similarity between them. So what is similarity? We took the Grantham matrix, which is an old matrix developed that's basically taking into account the composition, the polarity, and the molecular volume of amino acids, and has this distance metric. And this worked for us very well, actually worked better than Blossum or other matrices that are used for evolutionary comparisons. It's really the physical-chemical similarity of amino acids that we compare. And when we compare two k-mers, here shown for K3, we just basically sum them up for each position, like a mini alignment. And then to go a bit more into data, so we break down these sequences, we compute the scores between the, basically the average score between the two k-mers, but then, of course, you have lots of comparisons, right? These vectors are huge. If you just compare all k-mers to all k-mer, the signal is diluted, so that doesn't work. But what we do is we look at either the best match in these k-mer vectors or matches above a certain threshold. And this is basically the two type of SHARC scores we have. And to select them, we just select them empirically. And we also select the threshold empirically. So we take some kind of ground truth data we have from alignable still disordered regions. And then for each K, we can actually look at the performance, how it does in selecting similar sequences. And it doesn't do very well, right? So this is alignments with two different substitution matrices. And these would be the individual Ks with different thresholds or with different type of distance metrics. Google distance, Euclidean distance, you can compute many different distances. But what we can do is we can forget about these distances on their own and just actually train a classifier. Yes. So we can take these individual k-mer scores and we train a gradient boosted ensemble classifier, then will tell you basically if two sequences are homologous or not, by combining these different k-mer scores. And then we can also look at what it learned and which is the kind of contribution of each of these k-mers. We see that K1, this composition, is the most important to the decision making, but also K5 and 10, like shorter segments, they also contribute to it. And if we now look at the performance of our tool, then it looks like it's much better than alignments, in this case Hammer and Blast. And especially in this range of low pairwise identity, when usually you wouldn't even try to align sequences anymore, then it has a much better sensitivity and also F1 score. But how could you actually test if what you find, like these similar IDRs, if they are indeed let's say functionally analogous, for example? So for that, we looked in the literature of papers when people experimentally tested by swapping an endogenous IDRs into some other IDRs, like basically the entire IDR just chopped, and then look at if it retains function or leads to loss of function. So one such data comes from Alan Moses' lab when they took a yeast protein and they exchanged this IDR, I'm showing on top, with different yeast orthologs, for example. They align a little bit, but not too good. And it seems to be that the SHARC dive score for these orthologous proteins is very high, so it seems to work. They also took unrelated sequences that are kind of functional IDR swaps that don't align, again, very well, and still they have a high SHARC score. And then a swap that they did with a protein that did not complement functionality has a low score. So these are kind of results that give us some confidence that we get with these SHARC dive scores maybe makes sense. And now we have also a web server that you can use and just compute basically pairwise similarity comparison or search against the database. We have some pre-computed IDROMs of different organisms that you can search against for. Okay, so now we have a list of IDRs. We think they are similar. But now the question arises, what is actually similar and what is potentially the function of these IDRs? And for that, we would have to think about how is usually function encoded in these disordered regions. So they are often just flexible linkers, so probably not much more than maybe the composition would be conserved. But there can be also very specific molecular interactions or self-regulation or PTM sites, and we would actually expect certain short regions to be conserved. So we thought we could repurpose our algorithm, basically, and also look for which are the k-mers that are the most conserved, and find these SLIMs, these short linear motifs, like, for example, shown here, which I usually describe as like a linear expression. Here it's only 2 prolines and the positively charged residue that is needed, and this is enough to give binding specificity, let's say, to this domain. So we can take, again, a set of sequences from different organisms, run capture, and find these conserved k-mers that are shared. The output of the algorithm looks like this, so you can map back these scores on the sequence, and here, like, red color means this is like conserved, shared between many orthologs, and these would be binding sites. For example, in case of the SIC1 protein, these are known binding sites, and then we are happy that, okay, this seems to identify the sites that are known to bind proteins. We also wanted to test experimentally some new motifs, so we teamed up with actually Simon Alberti, who is working on the DAD1P helicase in yeast that has 2 terminal IDRs, and we identified a short motif, just 4 amino acid long, in the C-terminal IDR of this protein. Other tools, motif detection tools, also find it, but a longer region. So, just out of curiosity, we looked at the AlphaFold structure, what it predicts, and we were surprised to see that this short 4 amino acid long motif seems to form a tiny helix, and it actually binds the helicase core. And even if we run similar multiple AlphaFold calculations by perturbing the predictions, it always binds, so it's not just like by chance, but it seems to be always like highlighted here. Pink binds back to the helicase core, so maybe actually it modulates its function. So we actually deleted this motif, we mutated it to all alanine, and we also shuffled it. And all of these perturbations led to reduced ATPase activity in vitro, that is the native enzymatic activity of this helicase. So we think that now with these tools and with others developed also in the literature, there's really some cool other papers coming out recently, we can try to understand more what this disordered sequence space look like and have a similar picture. Probably doesn't look like this for IDR, there's maybe more overlap, what actually the sequence space looks like, and try to transfer information using homology. But I started to talk about IDRs, right, because I think they are actually important for biomolecular condensates formation. And when I started to think about this problem, I will not have here my usual introduction about condensates, since everyone knows that. We realized that for us, like for data scientists, we need actually very systematically collected data on biomolecular condensates. And back then, when we started thinking about it, we didn't know how many condensates there are in the cell, what is actually their protein composition. So we decided to build a database of biomolecular condensates. And when we started thinking about it, we realized we don't even know how to define a condensate. And we were discussing names of condensate. There was a lot of confusion. So we decided that we will build it in a community editable fashion. So we call it also the crowdsourcing condensate database and encyclopedia CD code. So it has this functionality that users can add their own information about their protein. It always has to be a PubMed reference, so everything has to come from already validated experiments. And this is the spirit that we wanted to do, and it's a collaboration with Viewpoint Therapeutics and Tony Hyman's group. So, of course, you can just search and use our database without contributing anything. So these are the most common search terms that people search for. As you can see, also this conference, highly represented FAST, for example, or TDP. We had a G3BP fund, for example. But we're also trying to stimulate the community to actually use it and add also information, because if you add your own paper, you know the best actually what you have done and you know what information should be shared. So we're trying to be active on social media. We have the condensate of the month, and we have also condensathons, we call them, when we provide free pizza to the students, and then for 1 to 2 hours they curate the database for us. So we basically have a list of papers have to go through, and that seems to work actually quite nicely. But so we had to make certain decisions when designing CD code. So for example, we decided we would split synthetic condensates and biomolecular condensates. And biomolecular condensates for us means that it was observed in a cell or in an organism. And we also decided to split the type of proteins we observe. So there are the so-called drivers, or others call them scaffolds or hosts, that are essential for the integrity of a condensate. If you knock this gene out, it's not there, or they don't form or dissolve. On the other hand, there are members or clients or guests, as others call them, that are part of condensates, but they can partition in and out, maybe as a function of stress or cell state, and they are not essential to the integrity of the condensate. So what have we learned so far from CD code, or back then? So we looked at the distribution of the condensate size. So we see a few condensates having lots of different protein members, like nucleolus and stress granules are one of the biggest, and there are some condensates that actually have a few members. But this might be just the bias of people labeling a few proteins like fluorescently, but actually not knowing what else is in those condensates, right? So this data usually comes from high-throughput mass spectrometry data and enrichment in these condensates, but we would need much more of this data actually to be able to believe in anything like that and understand it. But we also learned, and that was back then surprising, is the overlap between the proteomes of these different condensates is actually quite huge. So if you just look at the proteins that can be both in stress granule and P-body, there are like over 300 proteins that do both. So actually the specificity is not as great, and there's a lot of overlap, and it is actually quite difficult challenge for proteins to decide which condensate they belong to, so they can be in different ones. You can also test using this resource now if IDRs are actually common in condensate-forming proteins. That's kind of the paradigm in the field. So we can look at just what is the composition of IDRs in the human proteome for different lengths. I'm showing that. And then we can see what it looks like for CD code. And the difference is actually not that great. So there is, of course, enrichment. So there is more purple and pink. But there is 21% of proteins in CD code that don't have even a 10 amino acid long IDR, based on some prediction. So this is actually surprising, or maybe not that surprising after hearing also Karen's talks and others. So then we thought that, okay, this must be the clients. So they must be the ones that go in the condensate, but they are not important for actually forming the condensate. But then it turns out that there are also structured driver proteins, like the one that Tanya is working on, SPOP. So this is the crystal structure, and even if you look at the alpha-fold structure with pink, so the missing part is also predicted to be ordered. And there are other examples. This is a driver in nuclear speckles. This other protein I'm showing here, fully structured, is a driver in stress granules and a member of P-bodies. So there seems to be a whole range of driver proteins from almost fully structured to disorder that can drive condensation, and it's actually not a prerequisite to have IDRs. So seeing this, and also having this data resource, we thought we should also try to have a predictor that uses the sequence and predicts if a protein could form condensates that's not biased by thinking about disorder. Because these kind of second generation tools, like the one that also Thomas talked about, DPhase, they used mainly in vitro data, so LLPS kind of type of predictions they did back then. And also they assumed that disorder was one of the main driving force of condensation. So what we want to do basically to predict from the sequence is a protein part of a biomolecular condensate. We also thought it would be really great to be able to distinguish drivers and clients, and also be able to predict which condensate a protein would belong to. And then we gave up on this question basically because it turns out that proteins can be a driver in one condensate and a client in another. So that means that it's not only the property of the protein, but actually the collective of a given condensate. Of course, we could, again, predict an in vitro LLPS predictor, but that has been done, and it kind of works. So that we don't pursue anymore, but we work on a specificity that I will not talk about, but there is a great paper that just came out for GPS that does that, actually using the CD code data. It can predict white-wide nucleolar localization, and also which mutations, like which regions in the protein would contribute to this behavior. There's a new paper came out that does that. So one of the major challenges in any type of machine learning or data-driven approach is to design the training data. So we have the positive data now from CD code, and we are happy about that. But how do we define a negative data set? Like proteins that do not form condensates or are not for finding condensates because maybe they just haven't been tested. So the idea from my postdoc, Anna, was to look at the protein-protein interaction network of human and mark all the proteins that are positive, so known to form condensates. And then all of those that bind those proteins in this PPI network, say potentially in condensates, but they haven't been tested, So we say these are unknown, and we take as a negative data set only those that are at least 2 connections away from a known condensate-forming protein. And this also gave us a good balanced data set that we like usually in machine learning. So using this data, we trained a machine learning-based classifier that used actually sequence and structure-based features. It's called PICNIC, proteins involved in condensates in cells. Again, it will not predict in vitro phase separation. And the features that it's using is, of course, we use disorder and sequence complexity because it is one of the important features. But we also use sequence-based amino acid co-occurrence metrics within different distance windows. And this is basically like trying to simplify this like patterning of sequence of different amino acids like charges or hydrophobicity or tryptophans, which were shown to be important in the literature. And we also have this structure-based features, which is amino acid in a given secondary structure with a given confidence of alpha-fold prediction. And then, given all these thousands of different features, we let our machine learning classifier to actually select the most informative ones. So these would be some structure-based features, disorder-based features, and this amino acid co-occurrence-based features that actually the model selected 93 features at the end. If we just want to ask like which amino acids are usually contributing to the decision-making, just aggregating this plot basically, we see that it's lysine and tryptophan and also leucine, but cysteine comes up. So these are one of the top amino acids that appear a lot in this decision-making process. So we compared our tool, of course, to others and different data sets. Like there is another very good data set like FACET-TB that's similar to CD-code that we could test on the open cell data from the Chan-Zuckerberg initiative, and the CD-code kind of withheld test data that we didn't use for training. And the red line shows PICNIC, which seems to be performing very well compared to other tools. It has 80% accuracy in predicting condensate-forming proteins, so it's really not perfect. But it predicts another 1,500 new proteins that may form condensates in humans. So we teamed up with a postdoc from Tony Hyman's lab, Harry, who tested 24 of our predictions in U2OS cells, like expressed them. And we were happy to see that 18 of them, so the green circled ones, they form condensates. 3 of them were clearly not forming condensates, and 3 of them sometimes. So depending on a concentration-dependent manner, actually they did. So it's kind of, again, in this 80% accuracy. But what I want you to appreciate is that some of the proteins are fully structured, and they form condensates, and some, like in this case, disordered, and they don't form condensates. So again, we see, again, this pattern that is not so important, actually. Sorry, I will skip this. Yeah, so now we have PICNIC available as a web, like pre-computed for many species, also the Python package, and soon there will be a web server that you can compute it on any sequence. It can be a mutant sequence, a synthetic sequence, whatever. So what we can do now with such a tool, we can actually ask how many different condensate-forming proteins we predict in other organisms, so not just human, and ask what are the condensates across the tree of life. So if we go back to CD-code and just see what are the ones that we have already observed experimentally, there's a clear bias in mammals. Mouse, human, again, maybe just the bias of scientists studying these organisms much more, and also the relevance to certain diseases. But there are examples from viruses, from bacteria, as we heard yesterday, and they seem to be happening across many different species. So now with PICNIC, we can actually ask across any species, what do we predict? So when we look at mouse, human, and rats, then we predict around 40% of the proteome to be forming condensates. Then if you look at other model organisms, like, for example, flies and C. elegans, that it seems to be a similar number, and yeast, even more. These are plants, and then single-cellular eukaryotes, and then also E. coli. So we were very surprised to see that there doesn't seem to be a correlation with organismal complexity or having actually more condensates in more complex organisms. And this is surprising because we know that IDRs are actually much more common in eukaryotes. So no matter what metric you choose, bacteria and archaea will have much less disorder compared to eukaryotes. It was really a boom of acquiring these disordered regions. So according to this, if you look at the correlation with the percent of IDRs in a proteome versus the percent of proteins we predict to form condensates, there is no correlation. And it's lower path, so these are basically archaea and bacteria that have very low IDR content, but it still predicts that 40% of the proteins from condensates. So it's surprising. So it could be that our model is just wrong. and it doesn't predict it well. But since it was trained on human data, which is IDR heavy, I don't know, I'm surprised that it identifies all these proteins in organisms that don't have IDRs. And also some hope comes from the fact that we can actually validate like known data, because we have in CD code data for this species. Some of them have very little, but like, I don't know, in E. coli we had back then 6 proteins in E. coli, in CD code, and 5 we predicted correct. So that's one evidence. And also we have now collaborations with different labs working on archaea and viruses and so on, and they're testing these predictions experimentally. So now we have the tools to actually look at evolution of condensates across species, and that's what we do. And one of the focus that we are specifically look at is how are these condensates that seem to be functionally analogous across, for example, eukaryotes and bacteria related potentially, like nucleolus exists in all eukaryotes, and bacteria has nucleolus like RNP bodies. Is there any relationship or any evolutionary link between these, for example? So these are the questions we think about. How much time I have? 3 minutes? Okay. Then I just show you like one result about predicting sequence changes, right? So now that we can predict if a protein forms condensates, can we perturb now the sequence and predict if the condensation ability is changing? This is actually a very big task to ask from a predictor, right? It's basically predicting the effects of variants. So we tested how Picnic is doing in this task by collecting some data from the literature, and people took, this is the wild type, always as a star, and then there is some mutation that can be mutating, in this case, 13 residues, or deleting the IDR, no IDR, or doing some other perturbations to the sequence, and the red ones basically don't form condensates, and the orange ones like have condensates but with defects. So we basically want to see that the score is lower for the ones that are mutated and don't form condensates, and high for the ones that are still forming condensates. It looks very beautiful for the CBX suppurative. Here it doesn't look so great, but there is like a trend that the mutants tend to have a lower score. Of course, Picnic is not being able to predict just for one few amino acid changes that the score goes, let's say, below 0.5, which is a threshold, but there seems to be at least some trend in this direction. And we are working on a much better model specifically for this task at the moment. So to sum up, as a take-home message, I would say IDRs are not necessary for condensate formation. And we have evidence, let's say, from Karen's work yesterday when she showed that exactly that the helical region is the one that is forming, that is responsible for the condensation. And we don't see any correlation between the disordered and condensate proteome size. And condensate seems to be ubiquitous across species, and it's an open question how this beautiful complexity of condensates evolved. Last, I would like to thank my lab. I mainly showed data and results from Willis Chow and Anna Hadarowicz, and the CD code team. The current one is Maxim Sheremetyev and Ksenia. I don't have her name there. And the collaborators, Tony Hyman and Simon Alberti. And thank you for your attention.
Agnes, very nice. So this correlation between disorder and sort of organism complexity, it seems like well, maybe one thing to look at, and I'm curious as to whether you've done this, is to look at the strength of selection in these species, right? Because regardless of the organism's complexity, if there's very low selection, then we already know that they're going to accumulate tons of low complexity sequences and other things where selection is just too weak to remove them, particularly poly-Ns and poly-Qs, right? But they're like all sorts of different things. And it seems like you should see that. And that's one of the reasons why, at least I believe, many bacteria don't have these things, and why you actually see an elevation in, say, mycobacterium. Selection is really strong in E. coli, and it's really weak in mycobacterium. And so you expect exactly this sort of pattern. So have you looked at the basically, inverse of the effective population size as a predictor of whether or not you see these kinds of features?
Yes, but then, I mean, you would look at the... So I don't think Picnic is biased towards the sequences, so I think that's actually the reason why we don't see this correlation. Because it will not only pick up these low complexity regions or these repetitive sequences, but also the ones that are driving this bias structure. So, but I can still find evidence, like, look, yeah, I would be interested in seeing if there is any correlation. If we zoom in this region, of the very low IDR content if you still see the correlation with the, let's say, selection pressure.
Just a quick follow-up, right? So whether or not things form condensates is one question. Whether or not those condensates are quote unquote supposed to be there is another question, right?
I see your point.
Find things that are just sort of prone to form these things that selection is too weak to get rid of, but if they're still legitimate condensates that actually
I see. Okay, that's very interesting. Good look.
Agnes, really wonderful work, great analysis. It's quite remarkable that there's really no strong correlation or any correlation between disorder and condensate formation. I think that's something that comes very surprisingly. And I had a question regarding a comment you made at the very beginning, saying that the idea that condensates are linked to the origin of life is 100 years old. I was wondering whether the evidence that this is actually the case is any better now than it was 100 years ago.
Yeah. So since this is not my field, right, and we are not doing any of this synthetic biology, so I don't think I should comment on this.
Maybe you could.
Yes. But I think formation of protocells via these condensates is something that has been demonstrated, right? Like the actually go from a protocell to life it's very, very difficult. And of course that has not been demonstrated. But relatively complex protocells, I think there are examples in the literature.
Thank you.
Hi, super nice talk. I have a question in terms of features. So do you see any features emerging in those structural domains that you can, I'm now asking what you asked me 2 days ago, or yesterday. But do you see any features coming out from those structural domains in your analysis?
Yes, this is a very good question, and I actually had that question a few weeks ago in another conference, and I thought we should do this. So we should really systematically look. I mean, we do, because we stare at these structures a lot. We see something about beta structures, so that comes up as one, but let's say your protein would be like a coil type of interaction. So that could be also, I don't know, we should definitely do this analysis. I don't have answer yet.
Agnes, I have two comments. One is in the direction of simplification, the other direction is more complexity. And the first one we actually discussed on a nice sunny day a while ago. So I just want to repeat it, though, and to remind you that I know that aligning the sequences is very important, and I appreciate it. But is it possible to also do a data reduction where you assign a given amino acid, for example, a hydrophobic, hydrophilic character or charge, or uncharged character, and do the very same analysis, but then say, okay, maybe by doing this, we'll get some intuition about a more reduced parameter space, like the talk we heard yesterday with Z being a small number. That's my first question. And my second one is in the complexity direction. I appreciate and learn that not just disordered proteins form condensates, you have folded proteins, but not all condensates are the same. So it would be interesting, and I don't know how much data exists, to see if there's a correlation between folded proteins forming condensates that are very viscous or very elastic at short times, let's say, and IDRs forming condensates that at short times before they age are very fluid. And I think doing data reduction in the first question and data complexity in the second might provide intuition.
Yes. Yeah, just to answer on those. So the first is to coarse graining basically the sequence into something that's definitely a way forward also, and also learning something much more sophisticated, maybe deep learning is another option, right? The reason why it's difficult is because there's so few ground truth data on the similar IDRs that it's very, I think, easy to overfit on a certain type of behavior, let's say it could be charge or it could be hydrophobicity, but to really learn something general, we would need way more classification of actually IDR, similar IDR functions. But yeah, there should be a space that you can learn, which is more simplified than the 20 amino acids. So I agree. And then the other comment was...
Not all condensates
exactly
are the same, and they may have... I gave some physicists, viscosity, viscoelasticity may be different, I will guess. If you have structured proteins and if you have disordered proteins.
Yeah. So for the very limited FRAP that we did on the 24 tested proteins, that it seemed to be that the ones with IDRs are faster recovering in FRAP. So but it's not enough data, right? It's just a few points. But this could be something to look at, but there's not always FRAP measured on these proteins, right? Actually, we didn't record that information in CD codes. So yeah, I think to do that systematically may be a correlation indeed with the fluidity of condensates and how much IDR content is there.
Perhaps a silly question, but is the training of whether a protein is participating in condensates or not, is it somehow taking into account like the alkalinity or the salt concentrations in the cell, or is it all implicit that you see in that type of cell, it seems to condensate?
Yeah, so this is a good question. So we train only on the biomolecular data. So we think, okay, these are in cells, so there cannot be crazy concentrations of salt, or because the cell would not survive, right? So it's still within the norm of life, I would say. But there are, of course, differences. The same protein was shown to form condensates in HeLa cells, but not in HEC cells or so on. So there is, of course, differences coming from, it could be salt, it could be pH, it could be the other proteins that are expressed that are necessary. It's very, very complex. We all ignore that, basically. So we see if a protein formed in one condition condensate, in one cell, we call it as a positive. And it's also because it could be cell cycle dependent, stress dependent. So that's why also defining the negative is so difficult, because when is it negative? Like, how many conditions do you have to test that to call a protein negative? So, yeah.
I guess I'm asking this because of the archaea that you've been looking into, which are typically extremophiles, and then there's crazy salt concentrations, crazy pHs, and so on. So I guess, okay, it's implicit.
It's not part of the, no.
Thank you.
A question on different themes. I'm wondering, none of these methodologies you've introduced via K-mers are unique to proteins. And it's a large open question on trying to discover homologs for RNAs, where of course ground truth is much, much harder to estimate than what you have. So do you have some thoughts on whether these K-mer-like strategies that you explore, like whether is this like a unbiased way to start thinking about homolog detection in the absence of sequence similarity? Also ask because for paraspeckles, there's a common thing. The key RNA neat one has less than 1% sequence conservation, but still conservation in condensation/function.
Yeah, this is a good question. I mean, I know that for just aligning genomes, also K-mer-based methods can be used, like building phylogeny actually just from K-mers. So because genome alignments can be also complicated in the non-coding path. So it has been used for these purposes. Like it was mainly just Euclidean distance type analysis, so not taking into account similarities. I also don't know what would be the equivalent there for similarity for the nucleotides, right? Because I don't know what type of matrix you would use that, but just for whole genome alignments, let's say K-mer-based approach were successfully used. So I think probably yes, but then one would have to think about how to find, like, what would be this, like, coarse graining, or what would be the similarity matrix on the nucleotides that would be important for these RNAs that are important for structural context, right? Like binding proteins or forming RNA secondary structures and so on. Yeah.
Thanks. I was wondering if you see any themes in the proteins for which the predictions fail?
So from computational point of view, no, but we also tested negative examples. And actually we were surprised that many of them kill the cells. So they just don't express. And yeah, so there might be something in this negative data set that are just not expressing well, or they are toxic. But I don't know features. I mean, we could look at features actually also computationally, like what drives those decisions.
Thanks.
Okay, let's thank our speakers again. And,